Dear students, welcome to the class of basic genetics, and we are discussing about the gene mapping in prokaryotes. So today we are going to talk about the mapping of genes in phages, that is bacteriophage, right? Viruses that feed on the bacteria. So I would say this is the mapping of the rII locus is a little advanced concept, but I have included this topic for you at the base level so that you can at least have an idea how these complicated structures of genes emerges. And you know, in this class we will be mostly talking about the work of Benzer. He was a fantastic and fine scientist, and he did something again that, you know, it is very difficult for probably many others to even think about. And in one line, if I would say what is the significance of this class today, is that we will learn the fine structure of the gene. So I would say that this class probably should have come after the week 7, because the gene mapping started with eukaryotes, not in prokaryotes, right. So we learn about the recombination and crossing over, which I will be talking in my next unit. So probably just go through this class. If you have difficulty in following some concepts, you go through the week 7 and then probably come back to understand minor concept, although I am also not going into too much detail about everything in this particular class. But this is important for you to understand how we learn about the fine structure of gene. So the process of learning it was the genetic recombination in bacteriophages. So genetic recombination in bacteriophages was discovered during mixed infection experiment. So what are mixed infection experiment? Means you have a bacteria, right, and you are infecting it with two different types of bacteriophages. That is the mixed infection, right. So both the viruses are co-infecting one bacteria, and then you are studying the recombination between the two virus genomes. So in which two mutant strain, as I said, 1 or 2 were allowed to simultaneously infect the same bacterial culture. And how we do this kind of experiment? So when you design these kind of experiment, you need to have sufficiently high number of viruses infecting. Otherwise, what would happen? A bacterial cell will get one virus, another bacterial cell will get another virus, right, another type. Remember, there are two different viral strains. So then you will never have the chance to study the recombination between the viral genome. You have virus enough in the culture that there are high chances that both of them are going to infect together. That is one important feature. So I just mentioned here that one thing you need to remember, because of the intergenic means when two different loci are, we are talking when the recombination is happening between the two different loci, right. So then when two loci are in contact, then it is intergenic, we will say, and this was grand discovery that the recombination can even happen within the gene, means intragenic, right. So we will talk about it later. So Hershey in 1946 worked on two bacteriophage strains, and the bacteriophage type was even bacteriophage T2. So normal T2 plaques, what are the plaques? So when the virus feed on the bacterial cell, what it does, it lies. So you have seen in case of bacteria, there are colonies of bacteria, and what virus does, it lies those colonies. So if you have a plate and you grow the bacteria as a lawn, then what you would see as a result, small patches where virus has eaten the bacteria. So these are small patches, you know, these clear patches are I would say, I won't be able to draw it very well here, but these clear patches, right, everywhere the bacteria is growing, right, and then in the middle they are these viruses had eaten the bacteria. So these patches are called plaques, right. So, these patches are called plaques. These are small round areas are plaques, and you can, as you count the bacterial colony, you can also count the plaque on the bacterial lawn. So, what now they are telling in this slide I am trying to tell you is how people look at the mutant gene in case of bacteriophages, right. So, in this context, this Hershey was studying the gene R. So, the mutant R, the colonies are bigger. So, here the colonies are small, here the colonies are bigger. I will show you in a minute. And the other scientist, Salvador Luria, he studied another mutation, which is the name of that mutation was host range. So, host range means that a bacteria can grow in one type of, a virus can grow in one type of bacteria, but not in the other, but because of the change in the gene or mutation in the gene, now it can grow in another type. So, that is why they named it host range. So, this mutant has extended range of bacterial host, and wild type T2 phage infect only E. coli B, but not B2, but the H mutant actually can infect host B2. And the plaques has a center which is much more darker compared to H plus plaque. So, this is just a photograph I have taken from the textbook concept from genetics, and you can see that the wild type, so see R plus is the wild type, and the wild type colony size is this. And if you look at the mutant, this R, the colony size is this. So, the size is big. Rapid lysis, because of the rapid lysis, the colonies are growing bigger. Now, if you compare, if you have seen the R, now you look at the H. So, in the H, if you see that H wild type colonies are like these, whereas H mutant colonies are much darker, right. So, when these colonies are much darker and you have these H and R, both the mutants together, you can clearly see larger colonies with this darker center. So, what I am trying to say in this image, you do not need to follow every single step, but what I am trying to say, just by looking at the plate, you can identify the colony's genotype, right, that they are likely HR, and this is H plus R plus because of small size and clear halo around it, and then the darker brings the H mutant, and because it is smaller, then it is the normal for R, and the same is true for H plus R. So, but just by looking at the colony morphology at the bacterial plate, you can screen the likely genotype of the phages. So, that is very interesting. So, now as I just said, if you look at these kind of progeny and the parental types on the plate and you count them, you can see here, right. So, these are H plus R plus and H and R, they are the parentals, and these are the recombinant. Recombinants mean new combination where crossing over has taken place. So, the crossing over context I will discuss in the next class in very detail, but here you just understand that the recombinants are different from the parental type. Parental genotypes are like in Mendel we said, or maybe let us take the example of the dihybrid cross of the seed shape and seed color, and that is how we write both homozygous, both dominant and both recessive, and then the recombinants will be where the new combination, where one of the parental type phenotype is going with the other parental type phenotype. So, those are the recombinants. So, these you can see the recombinants are less and parental types are more, mostly happen in that case, and you can calculate the recombinant frequency by adding the total number. So, 12 plus 12 and then dividing by the total number, whatever the total number of this happens, and then multiplying by 100. So, this is the recombination frequency which would help you understand how close and how far the genes are. And so, again this concept in detail I will discuss in the unit 7, where actually it was discovered in Drosophila. So, you can, for now you just memorize it, and the next unit will help you understand to get the concept more straight. So now let's come on to the mutation, the analysis of the R2 locus. So this R gene I already mentioned over here can be identified because of its phenotype. So there are different classes. As I said, I have kept it very simple so that you can understand. There was a group of genes in the R1 group, R2 group, R3 groups, that there were different groups. So this R2 class of these a mutant in the phase 4, Benzer studied in much detail, right. So what he wanted to understand, he was considering this R as a locus. He took R2 class of that mutants, various type of mutants, where I will discuss the different mutants and try to understand those mutants to understand the structure of this locus, right. So R2 mutant shows altered host range, I did that this R2 has a specific. So R1 and R3, which we are not talking here, are different because they are growing in one type of bacteria only, but R2 mutants have this specific quality that they cannot infect E. coli K12. So remember, K12 usually written as K12 lambda because of the integration of lambda into its genome. So for simplicity we have just used E. coli K12. So R2 mutants could not infect K12 and they can only infect the strain B. So this whole structure, this idea, this concept that Benzer identified, that he can use R2 locus to map the gene's fine structure, because this can only infect one bacteria but not the other. I will show you how. Then that helped him identify the recombinants and different type of identifying the different mutants in context of one another. So what was his strategy? Let's go in detail. So he mutated. So if you have a gene, right, and you think that this gene is a unit, right, then you can think that it goes all together, right. There is nothing that I can break it even further, or there is another possibility that it can be broken down in small pieces. That is what Benzer was trying to understand, which one is possible. So what did he do? He mutated this particular gene at different regions by different methods, and he was having as many as 20,000 mutants with him, right. And then what he did, he had this gene with this mutation, the same gene in another mutation with same gene with another mutation at different position in the same locus, and he started testing these mutations with each other, right. And then he measured the recombination frequency, if the recombination, if there is any recombination between these two mutations or not, right. So if the recombination frequency is high, which means that these mutations are far apart. If the frequency is too low, then they are close to each other. So you can see the recombination frequency mapped mutation position within a single gene even, and two mutations in different site could recombine to give a wild type phenotype, that we come in a minute. So overall, so this line, don't worry about it, I'll talk about this assumption in a minute. So overall strategies that Benzer used to map R2 locus were three things that he did with the mutants, the 20,000 mutants that he had for R2 locus. Just for one locus he was having so many different mutants, and then he was trying to understand the fine structure of this locus. So he used complementation assay, deletion mapping and recombination. I will go to them, each of them one by one. So what is the complementation assay? So complementation assay is, complement means something is being given by one and the other thing is being given by another. That when you have one quality, you have another, your friend has, then you complement each other. He can write good notes and you can understand the concept better, you are doing studies together, you both are taking advantage of each other, right. So that is complementing. So let's look at the example. What did he do? He infected two mutants in K12. So E. coli stain just for the simplicity, the E. coli is not written. So, E. coli K12 was infected with both the strains together, and so if you infect both the strains together, you would have recombinants, right. So, then he recovered these recombinants, and ideally 2 mutant strains in a gene show a defect, right, and in his case what he saw, that they complemented. So, in case of E. coli, if you see and if you remember me mentioning before, this R2 mutants, different R2 mutants can infect E. coli B, but cannot kill the K12, right. So, ideally what you are expecting, if you are putting the 2 bacteriophages in the E. coli 12, right, in this E. coli 12, then these 2 mutants, if they fall in the same gene, they should not be able to do anything to this bacteria. But what if the 2 mutants can produce, let us say a mutant here and a mutant here, can produce a recombinant which is wild type, right. There are some mutants where they are both the mutants together, but then there are some viruses where they become wild type, and wild type can infect K12. So, I hope that you get the concept that what they are doing, they are using a K12 bacteria that cannot be infected by R2 mutants, but when R is normal, it can be infected by that. So, you can only recover plaques if you have the recombinant where a wild type is generated, right. So, that is what he was studying, and he was recovering the recombinant using this strategy. So, as I just mentioned, that complementation is each mutant strain provides something that the other lack, thus restoring the wild type phenotype. So, if the 2 mutants are at the same place, at the same thing, then they are homozygous mutants, right. But then if they are at 2 different locus and one mutation is only compromising one thing and another is another, then in combination you are getting both of it. So that is what I have written. Somehow this text is not being shown properly, but some pairs of R2 mutants complemented. So, as I have shown you here, the in text I just demonstrated, the complementation is each mutant strain provides something that other lacks, thus restoring the wild type phenotype. When many mutants were tested, each mutant fell in one complementation group. The 2 possible complementation group could be A or B. So, I will show you with a little example. Meaning is that if you have a mutation here, the another mutation is also at the same place, then you have the loss in both the copies, and they cannot complement each other, and so they do not grow. And because they do not grow, they both 1 and 2, they fall in one complementation group A, right. Let us say you have another mutation, one here, another here, right. Then although this is mutated, here you have the control, and in this case this one is mutated, but here you have the control. So, they can complement with each other. If this is 1 and this is 2, now you can put 1 and 2 in 2 different complementation groups, right. So, this is A group and this is B group, right. So, that is the example. I will show you once again just with more clarity so that you can understand the concept of complementation. So, some pairs of R2 mutants complemented and others did not. So, those complemented, they grew in K12. So, ideally they should not be growing in the K12, they only grow when they complement or they have the wild type phenotype. So, you can see I have tried to help you understand this concept of complementation and non-complementation by saying if you have gene 1 and mutation in one gene and another mutation in gene 2, right, then you have one wild type copy of gene 1 here and wild type copy of gene 2 there. And then if that happens, then your phenotype will be wild type and we call it complementation. And if you have mutation, both the mutation in same gene, very close place maybe, then actually you have non-functional gene 1 altogether. None of the copy is providing this product that we need from the gene 1, and then they are non-complemented. So complementing mutations are in different cistrons. Cistron, remember this word was introduced by Benzer. So cistron is what now we know that cistron is the gene, right? But what he said, which he very rightly concluded, that the cistron is the smallest functional genetic subunit, right, which is probably coding for a polypeptide. Now complementing mutations are in the same cistron that you can see here. So rII Locus actually have 2 genes. That was big, right? So a locus can have multiple genes, now we know, but then he discovered that even a locus can be broken in different sections which are coding for independent polypeptides. That was tremendous. So rII Locus has 2 adjacent cistron, R2A and R2B. And using this kind of complementation assay, he was able to divide all these 20,000 mutants he was having in 2 different complementation groups, and half of them fall in each cistron, right. So then he tested these individual complementation groups. So first he divided all the 20,000 mutations in 2 different groups, right. And then he said this is one cistron and here is another. Now he checked, so just conceptually what I am telling, all the mutation, let us say cistron A complementation group has all sorts of different mutations, some here, some there, some here, some there, and some actually are segment deletions, right. So some deletion means it's not just a point mutation, but a part of the segment is deleted. So these are deletions, and same is true for B. Now he started testing the mutation in this group to understand if even a cistron can get, mutations in the cistron can recombine and can create wild type phenotypes or not, right. Ideally we think that a gene should be non-divisible altogether. If it is non-functional, it is non-functional. But now we know that genes also have different segments, and if it's big enough, then, and if the recombination is being allowed, we can even allow the recombination within the gene. That's the discovery by Benzer, right. So what was his experimental setup? He co-infected E. coli B. Remember, E. coli B is something where R2 mutants can grow, right, and then allow the recombination. So in the E. coli B, he has co-infected the 2 different R2s and then allow them to recombine, right. They allow them to recombine and collected these recombinant, and after collecting these recombinant, what he did, he infected them with K12 in K12. So this one was B and this is K12. So in K12, now you remember I am repeating it multiple times, in K12 only those recombinants will grow that were wild type, where both the mutations were recombined out and you only have the wild type, normal functional copy of the gene, because the functional copy of the gene, if it has, then the virus can infect the K12 cells. And so what he did, by diluting serially, he was able to understand how much virus he has used to start with, and then out of that many virus, how many were actually growing on the K12 cell, to understand the recombination frequency. So mapping within a gene, now we are talking about intragenic. So R2A and R2B has hundreds of mutant sites. Recombination frequency between the mutation is equal to estimated distance. I already mentioned that, and detail will be in the next unit. Data showed genes are made up of small indivisible units, right. So Just a recap once again, because now you need to understand these two concepts. What is complementation and what is recombination? Complementation is when different mutations in different genes, right, and recombination is when the mutations, different mutations in the same genes or may not do the recombination. So, say if this is a gene A, you have two mutations in gene A, they can recombine and then you can get both the mutation on one and the wild type copy on the other side. So, this is wild type and this is mutant, right, and they are so close to each other, there is one possibility that you will never get the two mutations separated, they are always present there, right. So, they can recombine or they cannot recombine. So, this is the fine structure that Benzer was able to conclude by using these strategies, right. So, I am almost stopping here. So, for recombinations, he was also using the deletion mapping, as I just mentioned in my previous thing that he was having deletions. So, what he did, there are so many like 20,000 mutations, and if there are like say even 10,000 ideally, like maybe 5,000, then it is very difficult to check all these individual mutations one after the other, right. So, what he did is he used the deletions, overlapping deletions like that, and then point mutations. He brought the point mutation in the combination. Now, you just see that if this region is deleted and you are bringing a point mutation in front of it, which means that both the copies are now absent, you will not get the complementation, right. These genes will not grow, these viruses will not grow because of the mutation, and that help you put this point mutation in this particular region, right. And let us say another mutation still in the same region of the deletion, and that is how you can actually subdivide the point mutation in small groups which fall into the deletion area. So, using this recombination, complementation, deletion mapping strategy, he mapped this whole locus of one cistron, cistron A, you can see all the way here, and then cistron B is this one. And what these things are showing, how many mutations he got. So, you see there are some regions where he got so many mutations. So, he called these kind of regions as hot spots, because they are highly sensitive to get mutated, right. And then there are few where you do not find any mutation at all, there are some where there are only few mutations present in the region, even this A to D. So, with that I stop here, and this class actually helped you understand how we can even understand the structure of the gene. So, Benzer's recombination frequency was as low as 0.02, which means almost 2 base pair, 3 base pair, he reached close to that. He was able to get the recombinant between the genes that are so close to each other, and that was great discovery. Benzer did tremendous work in identifying the fine structure of the gene. So, Simon Benzer's work was revolutionary because he just proved one gene, one site idea. He showed that genes have structure and length. Recombination can happen within a gene which is intragenic, not just between the genes. And so, I will just show you this practical problem quickly so that we can see, I can see that how you can resolve the, how can you solve the problem. So, the problem is that we infect the E. coli with two strains, with the different strains of R2A mutant, right. So, this is mutant A and mutant B. After recombination, right, you bring A and B together, you plated it on E. coli 12 and you count the number of recombinant plaque. So, you total have 10,000 particles, and out of those 10,000, 50 were forming plaque, which means that there were 50 that are wild type. So, how would you get the recombination frequency? 50 by 10,000 into 100, just calculate it. And how they are close apart. If you get the recombination frequency, if it is in point and it is too low, then you can say that they are so close. I think it will come around 0.5 or 0.05, just check either it is 0.5 or 0.05, I think 0.5. So these two genes are very close to each other. So with that I stop here. I stop here with this question that has been shown on your screen. So you have two R2 mutants X and Y, and you perform two tests, complementation and recombination. And now in complementation you do not see any plaque that are formed. So what does it mean? And when you allow the recombination, you start seeing the recombinations. So the question is, what can you conclude about the location of these mutations? So do this problem. I have skipped this practical problem that I wanted to do because of the time limitation. It is also on your screen. You can have a look at it. You can solve both the questions and we can take it up in the activity section together. And so with that I thank you. I encourage you to solve these problems, interact with me if you have any problem, and thank you for your time and attention.